Welcome to Lesson 6 of Module 5 Heredity. In this video, we're going to be looking at polypeptide synthesis and proteins. And we're going to be answering the inquiry question, why is polypeptide synthesis important? So the order of the nucleotides in DNA determines which polypeptides are synthesized, and these are found in groups of 3 nucleotides, and we call these triplets. When a DNA triplet is transcribed into mature mRNA, the triplet is then called a codon. You can see that in our diagram here. So we can see that we have our mRNA through transcription process. We then translate that into a polypeptide. So here we have another diagram of how this process is working, and we see that the mRNA is a complementary copy of the template DNA strand, and the triplet codon does in fact tell us which polypeptide we are producing. So we mentioned in the previous video that there are 3 types of RNA, and we're going to go through their roles now. mRNA, also known as messenger RNA, this carries a complementary copy of the nucleotide sequence of DNA, and this specifies the amino acid sequence for a particular polypeptide. rRNA, or ribosomal RNA, works with proteins to form a ribosome, which is where the information in the mRNA is translated into a chain of amino acids. And we have our transfer RNA, which transfers amino acids from the cytoplasm to the ribosome, where they are joined to form a polypeptide chain. And the sequence of these nucleotides does then produce that polypeptide chain. So we can see this in a diagram here. We see the first mention of an important enzyme, RNA polymerase, which is important for that transcription to take place to form the mRNA. And we then see that mRNA move through the ribosome, which then becomes translated into a polypeptide chain. So there are a ribosome, and we do have our ribosomal RNA, and then we see our growing polypeptide chain as this mRNA moves through, and the transfer RNA is bringing in these codes, which then produces the relevant amino acid here. Again, another bit of a diagram about how this process works, and we can see that the 3 nucleotide sequence, or that codon, is really, really important. And we see the mention here of an anticodon, and this is the complementary to the codon being transcribed in the mRNA, and this anticodon does in fact bring the protein that we need, or the amino acid that we need, to form the protein. Here we have our table that we may have seen before, which allows us to work out what amino acid is going to be transcribed based on the sequence of the codons. And we can see that we have lots of different amino acids here, and we also have some codes that tell us to stop and start. The gene expression is really important, and this process is where the information stored in a gene is synthesized to form a functional gene product. And in eukaryotes, we see this happen in 3 stages. We have the transcription phase, which occurs in the nucleus. We have our RNA processing, and this occurs also within the nucleus. And when we see this export of this mRNA out into the cytoplasm, where translation does occur. In this processing phase, we look at exons and introns, and exons are regions of a gene that are expressed as proteins or RNA, and then we have our introns, which are our non-coding regions, and these are usually spliced out of the mRNA during this processing phase because we they don't code for any amino acids. Transcription in a little bit more detail, we do see the RNA polymerase be important in separating the DNA strands, and then when it unwinds, we see the mRNA, or the primary transcript, being created here, and then we do see our nucleotides attaching down here. Then we have our termination, and this is where it reaches a stop codon, and we see our RNA polymerase detach. RNA processing is important, and it only does occur in eukaryotes, as prokaryotes do not carry introns in their DNA. We see this process here, so there are lots of exons and introns within our DNA and then in our RNA sequence, and then once they're spliced out, we see our mRNA, and we only have coding sections here. Translation is when mRNA is translated into a sequence of amino acids and occurs on the ribosomes, and these ribosomes are docking stations for the tRNA to bring in the specific amino acid. If we compare the two processes in prokaryotes and eukaryotes, we see that the ribosomal subunits are different sizes. mRNA in prokaryotes only contains coding sequences for several genes, and then we see each mRNA contains a coding sequence for one gene in eukaryotes. There is no processing in prokaryotes, but there is in eukaryotes, and we don't see the adding of those features down the bottom. So why is this important? It's important to regulate it to make sure that we produce the correct polypeptides and proteins. And we can see here that it is required. So the amino acids then form a polypeptide, and then we have our protein, which is our fully functioning molecule that we want in our body. So these are just some key points here for you to read over. Thank you for tuning in for video 6. Make sure you tune in for video 7.